Okay, good morning, everybody, both online and in person. It's nice to see some familiar faces out here. Thank you so much for coming on a Saturday to spend a few hours with us. My name is Rachel. I am the nurse that works as the patient navigator for the University of Maryland PKD Clinic. Um, I just want to do a little bit of housekeeping, and then I will introduce our first moderator. So for those in person, we have a designated break where we can go out halfway through, have snacks, drinks. The bathrooms are just through these doors around the corner. Um, and the session is going to entail 4 expert talks. We have 2 in person and 2 virtual in true hybrid fashion. So we will be having both live audience questions and in-person questions that will be taken at the end of each expert talk. There should be about 10 minutes offered, give or take, for each talk. Following the 4 expert talks, we will have breakout sessions, 3 different topics. They will also be in a hybrid format. For those in person, we'll kind of disperse around the room. There is a Zoom link available for each breakout session virtually, and you can join the one of your choice. We have some fantastic people leading those, and I think that you'll get a lot out of it. 45 minutes is allotted for that time, with the official end time of the event to end at 1:30. So I would like to introduce Dr. Terry Watnick. She is a professor of medicine at the Division of Nephrology here at University of Maryland. She is a PKD clinic director, specializes in cystic kidney disease, and as a nephrologist and researcher, she keeps a full clinic schedule and has been involved in many research studies in ADPKD. So thanks, Terry, for doing some moderation.
Thank you, Rachel. So for our first speaker, it's my pleasure to introduce Dr. Greg Germino, who joined NIH NIDDK, which is the Kidney Institute, as the deputy director in 2009. He is also an adjunct professor of medicine at Johns Hopkins. He's the acting branch chief for the kidney disease branch and also the chief of the Polycystic Kidney Disease Laboratory. He is a longtime PKD researcher and will be talking to us today on PKD1 function and new paths to ADPKD therapies. Thank you, Greg. You can go ahead and share your slides there.
Thanks, Terry. And again, thank you for the opportunity to present to you today. And I apologize I can't be there in person. I'm going to try to pull up my thing now and see if it's going to let me do it. Oh my goodness, I'm surprised that worked. Anyway, thank you very much again for being able to present with you today. And I'm sorry I can't meet with you in person. So on this slide here, what you can see is really a unsettling statistic. This is from a paper on the published about a little over 10 years ago. And what's important here, what it shows is the age at which people started their renal replacement therapy from PKD, looking from 1990 through 2010. And so over the course of those 30 years, or over 20 years, sorry, while we had introduction of ACE inhibitors and angiotensin receptor blockers, which most people take if they have hypertension in PKD, what you can see is despite all of our best treatments during that 20-year period, the age at which people started dialysis didn't change over those 20 years. And so that's a kind of a sober reminder that no matter what we have been trying, it again hadn't really slowed much the progression of disease. And of course, now we have tolvaptan, and we've had tolvaptan now for over 10 years. And so I'm waiting for an update on the data to see whether this pattern really does shift to the right. And these curves now become, again, more over here in the 40s, 50s, and 60s instead of here in the 30s and 40s where it starts. So that's our hope, but we're clearly not there yet. So hopefully these slides are going to move. Here we go. So one of the things about ADPKD, as most of you know, is that it's in general, a very slowly progressive disease. So this offers both opportunities and challenges. The opportunity is that we have, many opportunities over time to intervene and give something that could slow progression, perhaps even reverse disease. So it's not a sudden catastrophe that if you don't catch the right moment, the disease, goes forward and there's nothing you can do about it. This really gives us a very long time to identify something and then start it. But the challenge of that, of course, is if we have to give something and we have to give it over a very long period of time, then the risks have to be very, very low for that therapy. So thinking about therapeutic strategies, there's 3 different kind of buckets that you can think about how we could approach the disease. One is to target downstream pathways, so those things that get dysregulated when PKD genes are broken. The second is to try to fix the mutant gene, the broken gene itself, or lastly, replicate the gene's function. So I'm going to go through each of these with a few examples over the next few slides. So thinking about targeting downstream pathways. So if you look at the ADPKD kidney here, as many of you have seen before, this looks nothing like a normal kidney. And so many people have looked at the tissue to see if we can get clues of what is causing the normal kidney to turn into this disaster we see on the right. And what people have identified is that there's an increase in the number of cystic cells. As you can see, these big holes, these are cysts. And those started out as parts of tubes that were maybe the thickness of a hair or a little bit more bigger than a hair. But you can see this is much bigger than a hair. The other thing that happens is the normal cells die. As you can see, this is all full of holes and scar tissue. There is no normal looking tissue, and that's all disappeared. These holes are filled with fluid, so fluid is secreted into the middle of these. There's a lot of scarring and inflammation, which again contributes to the pathology of the disease. So people have done a lot of studies on cystic tissue to try to identify what are those signaling pathways that are dysregulated. And on the right here is a cartoon of a kidney cell. In the middle is the nucleus. This is where the DNA is, and that's where we protect the genetics of the cell. Coming out on top there, that little finger pointing up, that's a primary cilia. All cells in the kidney, just about all cells in the kidney, have this little primary cilia, and it's thought to act like a little sensing hair and send signals into the cell to control cellular behavior. In blue and in yellow, in those boxes, are the names of different proteins or signaling molecules whose activity is changed in PKD tissue. In some cases, the levels are decreased, and in others increased. But there you can see there's many, many different pathways that are not working properly in cystic kidney cells. Now this is that same cartoon now with the green boxes. And in these green boxes are identified different classes of drugs that work on these pathways. So as you can see, there's almost as many drugs that could potentially be used to treat these pathways as there are pathways that are dysregulated. So there is theoretically, at least, a lot of potential targets in the cystic cell. One other implication of that, however, is that just stopping one of these may not be sufficient to slow cyst progression or slow cyst growth. Down at the bottom right here, where you see my pointer, these V2R antagonists, that's where tolvaptan works. It is blocking this receptor pathway here. And so it's blocking one of these pathways. But as you can see, one of the reasons why tolvaptan isn't a cure is it doesn't block all of these other pathways. And so there have been a large number of studies that have been testing different drugs or compounds that work on these pathways. The one in red, of course, is the one that you know about. That's the tolvaptan, and that one is the only one that has been approved by the FDA for treatment of patients and individuals with polycystic kidney disease. But as you can see, as you look down on both sets of columns here, there are many, many different compounds that have been tested. Most of them have stopped. They have not progressed further beyond the initial phase of studies. And each of these are targeting different aspects of signaling pathways whose activity is abnormal in cystic cells. Some of them did not go forward because they were not effective. Some, they had higher side effect profiles, so they couldn't proceed with them. They were not thought to be safe enough to give for a long term. And some might have been just, they've changed direction in their therapeutic direction for the company, looking at perhaps better targets. And so these are some of the trials currently in progress that are targeting, again, these pathways that are dysregulated. And I think Dr. Seliger is going to go over some of these in his talk that's coming up after mine. But I just want to point out a couple of them that are interesting. And this one here, this anti-PAB-A, this is an interesting new pathway that has been, 3 companies are targeting this. This is a protein that is present it's identified in the plasma of pregnant women. And by this protein is upregulated in cystic epithelium. and it's thought to activate growth factors to promote growth. And so I think there's a fair bit of interest right now in industry at trying to target that pathway with inhibitors. There's two listed on the slide here, and there's a third one that is in development. Then you can see on the right, people are also looking at dietary and interventions or supplement interventions that, again, might affect metabolic pathways. And I'll say a little bit more about that in just a few minutes. So there are a fair number of trials that have been done. There's a fair number, as you can see, that are in progress, and we're going to hear more about that in a few minutes. But there are still some outstanding translational changes. One of the problems is when we look at cystic tissue abnormalities, we're looking typically when we get human tissue, it's usually very late. No one's giving up their kidneys when they're still functioning pretty well. And so we're getting late stage tissue. And so when you look at tissue that's very, very, very late stage, it's hard to know what has happened directly from the protein being missing versus secondary or tertiary or quaternary effects. And so we could get very downstream, and we don't know when things are up or down, whether they're up because they're protecting the cell or they're up because they're driving the abnormal behavior of the cell. We also have very imperfect model systems for testing our therapies. The mouse models are good in that if you knock out the PKD genes, you get cystic disease in mice. But I don't know what 6 months of extending a mouse's life turns into with respect to human disease. If I slow cyst growth down for 6 months in a mouse, I'm making a huge difference in a mouse age. In humans, 6 months delay, is that making any difference? And so we don't really know how to translate that into human years yet. Other thing, as I said, is the side effects of drugs are often limiting, and that's why a number of therapies that looked like they might have had promise had to be abandoned. And then finally, and perhaps most importantly, we're treating the consequences of loss of PKD rather than treating the underlying problem, which is the broken gene or genes themselves. And so one of the emerging interests, of course, is can we fix the mutant gene? And so showing here is just the typical, the standard paradigm of how genes are encoded and made into protein. So our DNA, which is handed down to us from our parents, that is the blueprint, which serves then as the template for making RNA copies. Now, the RNA copies, they make many of them off of a single copy of the gene. And those mRNAs then go off and get made into protein sequences. So that's the translation part. ADPKD results from a reduction in PKD gene function. And so the mutation in a PKD gene, so a mutation here that we've inherited or that somatic that happened during de novo in a family, that mutation then results in either a broken protein or even no protein. It ends up with nothing here being made that's functional. And so some strategies that are under development to target this mutant gene include improving the mutant gene or protein's function, and I'll give an example of that in a minute, replacing the mutant gene and protein, and that could be like gene therapy, or finally, correcting the gene mutation. So I'm going to walk through each of these examples. So how about improving its function? So there are several strategies being pursued right now that might improve PKD1 function. The first is to boost the PKD1 and PKD2 protein expression. As most of you no doubt know, mutations in PKD1 account for about 85% of all ADPKD, and mutations in PKD2 account for just about everybody else. There's a few other minor genes that account for 1 or 2%, but very small percent. So PKD1 and PKD2 are the major genes. So there are strategies being pursued to boost their protein expression, because if you have a little bit, but it's not working too well, you can make more of it. You might be able to overcome that block. And the second approach is to improve mutant PKD protein function. And this is what I mean by that in the next slides. So can we boost PKD1 or PKD2 expression? Those are the genes that give rise to the mRNAs. So here's the gene, here's the transcription, and as I said, the DNA serves as a template to make copies, multiple copies of an mRNA. Now, Vishal Patel's group has identified a short piece of RNA that is made elsewhere in the genome that actually binds to the very tail end of the mRNA of PKD1 and 2, and it causes the RNA to be degraded, and it blocks the translation of that mRNA into protein. Now, why does it do that? We're not really sure. It tells us, though, that the level of PKD1 or PKD2 is pretty tightly controlled. The cell obviously doesn't want to have too much or too little. Too little, we get ADPKD. Too much, we presumably have other problems. So miR-17 is a naturally occurring molecule that reduces and controls PKD1 and PKD2 activity. But in the context of disease, if we have a PKD1 gene that it's making protein, but just not enough of it, or that's making protein that is only 20% efficient in its activity, what if we were to be able to make 5 times more of it? Then we could get back to closer to 100% function. And so Fischel's group has shown that you could make a blocker to that miR-17. And the idea is you block that miR-17, you keep it from binding to PKD1 and PKD2. And so now you have that RNA being made into lots of proteins. And in fact, there's a trial in progress right now that's looking at testing this. It's a phase 3 study that's just starting to look at this therapy, a therapeutic approach to treat ADPKD. So can we improve mutant PKD protein function? And what I mean by that is going to be shown on this slide. In the normal cell, and now what I'm showing here is a cartoon of just the top part of a cell. So down on that thing sticking up is the primary cilium. You can see PC1's listed there. And then down below are different structures in the cell that are responsible for synthesizing and then helping the PKD proteins get properly folded and then delivered to the proper place in the cell. So that's shown here in a cartoon in animated form. You can see PKD1 in red, PKD2 in yellow. They get synthesized and they traffic up the primary cilium to the cell membrane there. And that's where they sit. And that's the normal state. Now, in a normal state, where, for instance, somebody has what's called a missense change, that is where the DNA mutation changes one of the amino acids in the protein. So instead of it having just the right sequence, most of the protein's made, but it has a little kink in it. And so the protein may not properly fold. And that's shown here in this cartoon of this red PC1 that has bumps on it. So what happens when proteins may not fold properly is they often end up in the cell's trash can, the proteasome. So they get degraded, and occasionally a few will get up to the surface, but mostly they don't. And so there are strategies that have been used successfully for other diseases, like cystic fibrosis, and now being considered for ADPKD, where one makes a drug, and the drug goes to the mutant protein, corrects its folding, and now allows it to, instead of go to the garbage can, it goes up the primary, it can go up to the primary cilium, as shown here. And so this now normalizes, or at least gets closer to normalizing, the amount of functional protein that it gets delivered in the cell to do its business. And in fact, Vertex, the company that developed the drugs for cystic fibrosis, is now testing in ADPKD whether they can use a corrector protein. So again, that oval I just showed you, can it bind to PKD1? Can it help it fold better? And can it get out of the ER and out of the other structures so that it doesn't go to the garbage can and instead goes to the right place? So, of course, one of the other great strategies that people like to think about is what if we could just replace the mutant gene in protein? And that's considered gene therapy. So if on the cartoon on the left here, you have a cell, non-functioning gene, you introduce the functioning gene, and now you have a cell that's functioning normally. And so that is the principle. And in fact, Steve Somlo, in a very elegant study a couple of years ago, showed that theoretically this could work. In his model, what he's showing here on the right is a mouse model of PKD2. And so in this model, they turn the gene off. As you can see on the top, as the animals age, they get very cystic kidneys. And on the bottom right, you can see their curve. This is a measurement of kidney size. The higher the number here on this axis, the bigger the kidney. And so you can see they were able to get animals to have kidney disease and then they turned back on the PKD2 gene. And what happened to the animals who had the gene turned back on is the disease got better. It corrected. Those animals that did not have the gene turned back on continue to get worse PKD. So this suggests not only can you stop the disease, but if you get it early enough, you might even be able to reverse at least the early stages of disease. So that sounds great, but the problem is with ADPKD is that the challenge is getting the gene to the correct cells. The PKD1 gene, as I'll show you in a minute, is very large. And most ways we deliver genes to correct for transgenic therapy to do a permanent correction is usually using a virus. And those viruses, they take out the viral DNA and they put in the DNA of interest, but they have a size limit. And that size limit typically is about half the size of PKD1. So it can't get in there. There is an approach using a short final segment of PC1 that I'll comment on in a few minutes that potentially could fit. And so there's people looking into that. The other problem is that getting this gene into the kidney and bile duct cells is not a trivial problem. And this is true not just for ADPKD, but for any genetic kidney disease. And a second problem, of course, is getting the correct expression of the gene. As I said before, we know that the disease results from too little expression. So whether you break it by a mutation or you break it by some other way, you're going to get cystic disease. But if you have too much, we think that could also be a problem. There was a study that suggested if you make way too much, you can also get a cystic disease. And so, and we know that the body has developed systems to keep that level of PKD1 expression in adult kidney pretty controlled. So getting that level right might be a challenge. And then finally, making sure that once we get it there, it stays being made. There are delivery systems that can give you transient expression, short-term expression. I can put it in a little bit of packet in a little bit of lipid, a little bit of fat, and you can send that off to cells, and it'll take it up, and it'll stay there for a bit of time, but eventually it goes away, and you have to then repeat the treatment frequently. And then finally, if you use a viral system, and it gets into the DNA so it stays forever and you don't ever have to treat again, you have to make sure that where you put it doesn't affect other genes in the genome and cause some other disease problem. So what are the other things that people have talked about is, can we do gene editing? Can we correct the gene mutation? And that is possible now because of a technology called CRISPR. CRISPR was the result of actually fundamental science. Somebody was looking at infectious diseases and bacteria and viruses and looking how bacteria change and modify their DNA. And they came up with a system that turns out to be extremely effective for using for human cells and animal cells, this CRISPR-Cas9 system. And so what the CRISPR-Cas9 is a set of proteins that have the ability to read the DNA and bind to the DNA, and they have a guide RNA, which is kind of a copy of the sequence where you want this CRISPR-Cas9 to sit down and bind. And once it finds the right place, it cuts the DNA. And if you give it a copy of the normal sequence there, a short copy of it, it can then rewrite the DNA, correcting the mutation. So gene editing, again, would be terrific. You could just keep your gene yourself, your same gene, just fix the mutation. It's got to go into the repairman, he fixes the mutation, and off you go, perfectly healthy. So this is the holy grail of corrective therapy, but it is still a bit in the future. It has some of the same challenges as gene therapy does. So it brings me to the last bit of the talk, which is, if we can't easily bring the gene back and we can't easily fix the gene yet, what could we possibly replace the mutant gene's function? If I know what PKD1 and PKD2 do, could I then do that activity in some other way? And so that's what I'm going to spend a few minutes talking about, and approaches that my lab is taking to try to solve this problem. So shown on this cartoon on the left is a schematic of what the polycystin 1 protein looks like. Polycystin 1 is the protein encoded by the PKD1 gene. PKD1, as I said, is a very large gene, and this is a very large protein. If the average protein in the body is 500 to 1,000 amino acids in length. PKD1 is 42 over 4,000 amino acids in length. So it's over 4 times to 8 times larger than the usual gene. And so it has a lot, so it's a very, very, very big gene to work with. And that's why it's hard to put it into viral delivery systems. It also has a lot of interesting properties, as shown here. There are these arrows that you see, one here and one here. These are places where the protein is cut. And these cut pieces have different functions. And we're trying to decipher what those functions are. But some of them seem to be essential to the function of this whole PKD1. Now, PKD2 is shown here on the right. This is it's a much smaller protein. And it looks like it might have channel properties. And what I mean by that is it opens a hole like a gate in the cell and allows charged molecules to flow into the cell. And so it's thought that the polycystin-1 and the polycystin-2 proteins are partners. They have to work together, generally, at least in the kidney. And together they control a gate that allows molecules, charged particles into the cell. But as I show on the slide here, there is far more we don't know about these proteins than we know about these proteins. We don't know what goes through that opening they make, that gate. We don't know what makes the gate open and close. We don't know what those particles do once they go through the gate. And we don't know, as I said, what these particles do, these parts of the protein, when they get cleaved, what they are really doing. And so my laboratory is focused on trying to determine the function of polycystin-1. And so I'm going to go through just very briefly some of the methods we use. So we do a lot of cell culture. And what that means is we take cells. They could be kidney cells. They can be other cell types. And you can grow them in a laboratory on a dish with just media. And so when we've done some of these studies, one of the things that we discovered is that last piece of polycystin-1, that arrow that I showed you, that is that little bit that's inside the cell, that goes off to the mitochondria. What I'm showing you here on this cell are cells that are colored. Purple is for mitochondria, and green is the PKD1 piece with a tag on it. On top here are mutant cells. On the bottom are cells where we've put back in just that little piece of PC1. And this here, what you see, looks like worms. That's the corrected mitochondria. So that's what mitochondria should look like, not like this. This is what they should look like. And so putting this polycystin back in corrects mitochondria activity and may partially rescue its activity. And in fact, that's partly why there are studies underway now to see if this CTT, this very small piece of polycystin-1, is able to actually delay disease in models. Michael Kaplan's group has shown in animals that it seems to do that quite well. So this is what we've learned a little bit just doing cell culture methods. We can dissect out specific portions of polycystin-1 and test their activity. As I mentioned, we've made a lot of mouse models and others have made a lot of mouse models. Here's an example of our polycystin-1 conditional mouse that we made over 20 years ago and still use, and many other groups still use. As you can see, this looks an awful lot like a human ADPKD kidney. So using this model, we and others have done a lot of studies trying to understand, again, the pathogenesis and the pathobiology of disease. But one really wild result that we came across a number of years ago is that we found that if you turn PKD1 gene off during the first 2 weeks of a mouse's life, they get cysts within days. If, however, so if I turn it off at day 13, they get cysts quickly. If I turn it off at day 14 or 15 or any time thereafter, it takes a very long time for them to get cysts. And so we're interested in trying to understand what it is about that window can give us insight into what are the other factors that are required for cysts to form. Because if we can understand why it takes so long here, we might be able to even push disease even further out. And so these are sorts of studies you can do with mice. And then I told you already about Steve Somlow's study where he's able to turn PKD2 back on and rescue the mice. So these are, again, studies that are giving us some insight into PKD1 and PKD2 function. One of the other approaches that my group and others do is it's called transcriptomics. This is the process by which we take tissue or cells and we examine all of the genes that are being expressed under normal conditions and abnormal conditions. So we can collect the RNA as shown in the bottom left, we sequence it, we compare the gene activity, and then we see what's up or down in normal versus cystic samples and cystic cells. And that gives us ideas of different pathways that might, again, be regulated by PKD1 and PKD2. And it was studies like this that told us that loss of PKD1 results in impaired fatty acid metabolism by mitochondria. So again, we saw this PC1 goes to the mitochondria. We found by transcriptomics, again, that PC1, loss of PKD1 results in impaired mitochondria activity. And so collectively, it gives a picture that the metabolism of cells with PKD is different, is regulated differently. A third model or third approach we use is to try to understand the proteins, so the partners, the friends, collaborators of the PKD1 gene. And the strategy we've used is shown here. We've made a mouse where we've introduced special tags in situ to the polycystin-1 gene, to PKD1 gene. And we can use those tags to isolate polycystin-1 and purify polycystin-1 from mouse tissues. And the strategy is shown on the bottom there. So we take the kidneys or the other tissues, we grind it up, we make like a soup. We can then use beads to pull out polycystin-1. We wash it, and then we can use a special machine to determine not only polycystin-1, but all the other proteins that were sticking to polycystin-1 when we isolated it. At top right, you can see we get some of the friends of PKD1, and again, we identified protein-protein interactions linking PKD1 to mitochondria and metabolic functions. But one of the problems with the transcriptomics, the cell culture, and the proteomics is you get too many targets. So this is actually from our study of polycystin-1. And you can see that all these little blue dots identify proteins, and the black lines show how they connect back to either each other or indirectly to polycystin-1. And so we have this huge, gamish of proteins that are somehow associated with polycystin-1. So trying to figure out which one is the most important can be really challenging, and particularly if we have to use animal models, which take a lot of time and use a lot of animals. So we've complementing this and supplementing this with a new approach using organoids, and I'm going to show you what that looks like here. So there's a couple different ways that we've been able to make organoids. So an organoid, starting on the far right, is like a mini kidney. It's not a kidney, but it has a lot of the kidney cell types all linked up in a similar way as they would be in a real kidney. But you can grow this in the laboratory. You don't have to have an animal to do this. It starts out with taking cells, either from embryonic mouse kidney or taking pluripotent stem cells that can be induced to make progenitor cells. So these are like the cells that will give rise to a kidney. We can treat them with a bunch of different chemicals, and now suddenly they go from being completely a cell type that can make almost anything into kidney cells, kidney precursors. And we culture them a little bit longer, and we can then make what's shown on the right, which looks like a little kidney organoid. And so this is what it looks like in real life. So plotting here, these NPCs, these are progenitor cells. So these are the cells in the mouse that would give rise to a kidney. And as you can see, when we just put them in a culture dish and we treat them with various factors, you can see they start to change. They start to differentiate, and they form tubules. They form little glomeruli, at least they form what looks like a glomerulus without vessels, and then they form tubules. This is what it looks like if you stain it with markers, just to show that it's not just balls of cells. These are different markers that identify different types of kidney cells that in the normal kidney. And as you can see here, we see cells from many different lineages lined up and linked up like they would be if you were looking actually at a mouse kidney. And so one of the advantages of this is that these form quite quickly. I can go from just back here, a ball of cells, to here on the right, day 13, what looks like a kidney in less than 2 weeks. So it's a very rapid way to do it. And you can do this in hundreds of these at a time. So you can use it for screening all sorts of things. And so this is what happens when you take cells from an animal with PKD1, where it's been where the gene can be turned off. On the top set of organoids, we left the gene intact. In the bottom organoids, you can see where we had turned PKD1 gene off. And you can see by 2 weeks of age, actually by day 9, day 10, we are already forming cysts. So this is a very, very rapid process of making cysts. And why we think this is particularly useful and powerful is that in animals, when this happens, it's extremely difficult to catch that window of time. When we do it, it's a snapshot in time on any given animal. There may be cells that are early cystic. There may be cells that are now late cystic. It's very difficult to capture it at the right exact window. In this model system, it's completely, it's completely reproducible. They regularly make cysts by day 9, day 10. And so we think we can use this system to understand what happens as cells transition from normal to cystic. I would look at it kind of thinking like the Big Bang of the universe. We're here right now, right? This is our galaxies. Here we have cysts. And okay, we know what the cysts are right now, and that's great. But I want to know the first stages. What happens when you first turn the gene off before it actually becomes a cyst? And you have all these other pathways that get dysregulated. And so with this model system, we can go back in time, just like you can with the telescopes, going further and further back with these various space telescopes to just go back to the earliest time before the Big Bang. And so we're doing this sort of approach now with organoids, looking very, very intensively at each of these phases to see if we can identify the transition states and what are those factors that are first off when the PKD gene is first turned off. So just to end, short term, I think most therapeutic options likely will come from treating altered downstream pathways or boosting PKD expression or correcting the effects of missense changes that reduce their function. These are treating consequences and rather than treating the underlying problem. So they may delay, and maybe they'll delay enough to keep people healthy through the rest of their normal lives. That's the goal. That's the hope. But many of these may be most effective for those with milder disease who have less need for life-saving therapy. I think true cures, based on gene therapy or gene editing, will probably take some more time as we work out efficient and safe delivery systems. Although I think there is some exciting progress in that space. I think replacement of function, once known, could potentially provide some similar benefit. With that, again, I want to thank you for the opportunity to present with you all and to talk with you all today, and I'm happy to take any questions.
So I think we just have time for one question.
Yes, I'm Paul Scribner. I'm with the PKD Foundation National Office. And my question for you, it's clear to me there's a ton going on right now in the PKD research space. I've seen it directly, and your presentation was very clear and really helped me understand it even better. But I wonder, from your perspective, what is the most important things that the foundation can do to help accelerate the time to get these trials fully enrolled as we move forward and support and accelerate the progress toward these better treatments and a cure? I mean, if you were to provide your perspective.
So I think the one important role for the foundation, or a couple different important roles. One is to, again, continue to support, pilot studies as you do. I think that's really important, having that research fund available. I think a lot of we need fundamental research to help get us those targets. But I think the other thing is, as you're doing, is making people aware of studies and the importance of participating in clinical studies and clinical trials, because there's a limited number of individuals, and yet there's a lot of targets. And so we have to think about how we can prioritize our different clinical studies and by the more people we have participating, the more different therapies that can potentially be tested. Dr. Seliger could probably also speak to this even better, because he's actually on the front lines of testing these things with people.
So with that, I think we're going to move on in the interest of time. Thank you, Greg, very much. That was comprehensive talk.
Thank you.
Okay. So our next speaker is my colleague, Dr. Steven Seliger. He's an associate professor of medicine in the Division of Nephrology here, and also adjunct faculty in the Department of Epidemiology and Public Health here at the University of Maryland. He's a leader. He is the principal investigator of our clinical and translational core, which is part of our PKD Core Center. And he's a nephrologist and epidemiologist and has been involved now in numerous PKD clinical trials and is our clinical trials expert here. So with that, Steve, I'll turn it over to you.
I want to start by posing the question is, why should somebody participate in research? And we heard a little bit from Dr. Germino about the importance of that, and of course what the PKD Foundation does to help connect people to study opportunities. So I just wanted to go through sort of 3 theoretical or conceptual benefits for participation. And the first maybe is the most obvious, which is the direct benefit. So you'll see that most of these trials involve the random assignment, often to an active study drug or a placebo, a dummy pill or sugar pill. And of course, if you participate and you're assigned to the active study drug, and the active study drug turns out to be successful and effective, then of course you gain the advantage and the benefit from taking that medicine before it's maybe years before it's even available to everyone else. But even if that drug turns out to not be effective, and or B, you participate in the study, but you are randomly assigned to the inert, to the placebo, there are still many potential benefits. And those come from more careful disease monitoring. Often that's something that's built into the research study protocol or procedures. For an example, accurate measurement of total kidney volume, I'll get to that in the next slide. That is an essential part of understanding one's risk of progression if you have PKD, and it often is very hard to obtain in a standard MRI. But the research studies provide this almost all as a matter of course. Then the last one maybe is potentially or conceptually from the population standpoint the most important, which is you're contributing to fundamental science and understanding that will be of benefit to everybody with polycystic kidney disease, both living now and for generations to come. And so, of course, the development of these medicines and the treatments is entirely dependent on people volunteering, right? And nothing can occur or can progress without that. And the perfect example of that is the development of the drug tolvaptan. And some of you in person and online maybe were volunteers in the studies that led to the approval of tolvaptan as the very first drug approved by the Food and Drug Administration to help people with PKD. That took more than 1,000 patients, thousands of patients around the world. So none of this is possible without people volunteering, absolutely. So the next question is, who should take part or who could take part in clinical trials? And this is where I think gets to who's eligible for trials. I'm going to get to that for each trial I talk about, but as a general principle, those who have the greatest risk of progression, going on to progress to kidney failure, and the lowest risk of potential side effects for a given medication, of course, those should be the people who will be invited or selected to participate. And I did want to comment on some methods to assess the progression risk. The most commonly used in our country is something called the Mayo imaging class. Many of you have already may be familiar with this. It is simply a classification of one's kidney size or kidney volume for your age. And so this graph here shows the expected rate of decline of kidney function on the y-axis, estimated GFR. And in the black line, the x-axis here is just years from the time that it's been measured. And so this is a black line is people without polycystic kidney disease. All of us, as we get older, our kidney function will decline a tiny bit every year. This is part of aging, natural aging. This green line is people who are Mayo imaging class 1A, the smallest kidneys for age. Bigger kidneys than normal, but the smallest among everybody with PKD. And you can see that they also decline, but at a rate no faster than if they didn't have polycystic disease. In contrast, in the red line is 1E, the largest kidneys for age. And you can see that in a period of only 10 years, even starting with normal kidney function, that they are predicted to go on to kidney failure. So this turns out to be a very powerful tool, and it's actually quite easy to get at because you only need these two inputs here. There are other tools which I'll skip forward in the interest of time, but the Pro-PKD score, you may come across this. It's used largely in Europe because it depends in part on genotyping and some other clinical information. Note that being a man gives you a higher risk for developing kidney failure. This is true in polycystic kidney disease and other acquired kidney diseases as well. There is a new model, I'll just let you, just to throw this out there, which was published about 6 months ago. This model for prediction seems to be more accurate than the others and was derived using really critical federally funded studies. This hasn't yet been fully validated or implemented into clinical trials, but certainly these kinds of models might be. Okay, so one approach to testing drugs, and this is one, again, that Dr. Germino alluded to, is to repurpose drugs that are already on the market. This is a cartoon very similar to what Dr. Germino described, and I find it overwhelming as well, even though I've been in this studying PKD for 15 years. But in black here are examples of drugs that target all these different pathways. I won't go into detail on this, but just to let you know that you can see where there's different possible ways to intervene in the disease process using drugs already available. So the advantage is that these drugs have already been developed. They're in some cases already, or in all cases, already approved for treating other diseases. So we know a lot about how to dose it, how the drug is handled in the body, what are the side effects, what its safety profile is like. They can, because they're not on patent, they can easily be used also to study mice with experimentally induced polycystic kidney disease. And as I said, the side effects are relatively well described. So this is an example of drugs that are being repurposed, and I'm actually going to go through these, most of these in more detail. The one that I wanted to touch on very quickly on this slide is the diabetes drug metformin, and many of you may have read about it. It's like people are using it or thinking about it to improve longevity and treat diseases of aging. It is a drug that has been around for decades to treat type 2 diabetes. It's very safe. It targets some of the same pathways that are involved in polycystic disease. So in mice, if you give them metformin, you can slow the cyst enlargement, improve or slow the decline of kidney function and lower blood pressure. So Dr. Watnick and I were part of a multi-center phase 2 trial, which looked at metformin in polycystic kidney disease. And some of you here may have been part of that trial. It was called the TAME-PKD trial. And we found that actually the drug was safe and well tolerated. Some of the potential side effects we were concerned about its use in kidney disease, we didn't see any of. So that was really great. And now there is a much larger trial underway. Unfortunately, not in the United States. It's underway in Australia, New Zealand, and in Europe and the UK. But this drug is pennies a pill. So if it is proven to be effective, it could be quite helpful. Let me go through some of the other drug categories. The one I wanted to talk about next was the SGLT2 inhibitors. So these are drugs like Jardiance, Farxiga. So it's hard to watch TV now without seeing ads for it. These were drugs that were developed to lower blood sugar, and they do that very well. So it blocks the reabsorption of sodium and glucose in the tubule of the kidney, and that ends up actually. So you think, well, what does that help? Well, the first thing it helps is just to get rid of more sugar from your body, so that's great. But it seems to also reduce the pressure inside the filters of the kidneys, and to reduce this fibrosis and scarring, also to reduce blood pressure as well. All those things that we know can help long-term kidney function. And so in patients who have chronic kidney disease, not from polycystic disease, it lowers the risk of kidney failure and progression of chronic kidney disease. And this is one example of the trial, which was using, dapagliflozin or Farxiga. Note that this was a study in 4,000 patients internationally, and the risk of going on to kidney failure was substantially lower, so 9% versus 14.5% in the placebo group. So it lowered that risk on an absolute scale by 5%. So this drug and other drugs like it, Jardiance is the other one, are FDA approved for the treatment of chronic kidney disease, not from polycystic kidney disease. What about polycystic kidney disease? There are complex reasons why none of those trials included polycystic kidney disease. I won't get into that for the purposes of our discussion. So what do we know about its effects in polycystic kidney disease? we're going to know a lot more in just a couple months, and I'll tell you why, from a trial that Dr. Watnick and I are doing. But we have some data from observational. So this is, again, not from clinical trials. These are databases of people who were taking this drug and some who weren't, and to see what happened. In this case, this is a very nice paper from the National Veterans Administration. I work for the VA as well. These are people who had polycystic disease and also diabetes. So some were getting these drugs for diabetes, not for their kidney disease. And this shows what happens to their kidney function. This is on the y-axis here. Before they got the drug, their kidney function was declining, which unfortunately is what happens in polycystic disease. And once they started it, it seemed that the kidney function stabilized. Yeah, so not getting better, but not getting worse, which is, of course, would be an enormously successful goal. There was also a small trial published in Japan, which only looked at 6 months of the drug. So obviously, shorter duration than one could say whether it's effective or not. But it also seemed, this is the black line, was the line that got Farxiga, and the dashed line was the line that didn't. And these patients were already getting tolvaptan. So it suggested, again, there's some kidney function decline. This is not a large difference, but it's only 6 months. Very preliminary, but somewhat suggestive that there might be a benefit. So there are multiple trials ongoing using this drug in polycystic disease. This is the trial that started and finished first. It's also the trial that I'm co-leading with a colleague at the University of Colorado, and Dr. Watnick is a co-investigator in the study. We treated 50 people with placebo or empagliflozin, that's Jardiance, and we treated them for 12 months. So the last person just finished last month, and this will be really the first in polycystic disease trial to look at a year of treatment, and we hope to have results very soon. As you can see, there are larger trials with hundreds of patients for 2 or 3 years. These are going on in Europe, not in the United States at least, and they probably won't have data for a good 5 years or so, but we're hopeful that all of these together could someday show that this drug is helpful, or whether it is helpful. And these drugs, dapagliflozin or Farxiga, is just going off patent. So again, by that time, these drugs should also be pennies a pill. I've had some patients ask me, should I just take these drugs because they work in other kinds of kidney disease? And this is what I would say. These drugs are not approved in polycystic disease. The FDA made that very clear. The current consensus treatment guidelines advise against their use for the purpose of slowing disease progression. So this is the consensus guidelines that came out last year, international consensus guidelines. And Dr. Watnick led a group that published a series of commentaries for the U.S. National Kidney Foundation on these. So the practice point is here, you should not use these drugs to slow kidney function decline in PKD. And studies are needed to evaluate the safety, efficacy, and effects. Well, we finished that study. I hope to have the results very soon, and then the longer-term studies will follow with that. So that would be my answer to someone who's asking about it. Now, if someone happens to have diabetes is really common, also affects people with PKD, to use it for sugar lowering, that's a different question, which this guideline doesn't address directly. I wanted to talk to you about another drug that is being repurposed, and which we are part of here at the University of Maryland. This is a drug called bempedoic acid. You may not have heard of this drug. It's not really been heavily in the news or anything, but it's been around for years. It's an FDA-approved drug for people with high cholesterol. Now, you might be thinking why most people with high cholesterol already take a drug. They're called statins. So this is a drug for people either who are already taking statins and their cholesterol is still too high, or, can occur in a minority of people who take statins that can't tolerate the drug because of side effects. But it acts on an enzyme within the cell that in PKD, that same enzyme promotes the growth of the cells lining the cyst, which eventually leads to cysts and enlarging cysts. So the drug also is interesting properties for the use in PKD, which is the drug is inert in the form you take it. It's taken into the body and activated into its active form only in the liver and in the kidneys, which of course are the two organs most affected by polycystic disease. And in mice, you can give this drug and it slows cyst enlargement and it reduces scarring and fibrosis. And this is a cross-section of a mouse kidney in some studies performed by the investigator that we're working with on this trial. And you can see this is a mouse that has polycystic disease and was given just placebo, and after 60 days, and this is the one that got the drug. These holes here, as you can see, are cysts, and this kidney is much larger than this. And after 120 days, the differences are even more marked, right? These are highly cystic kidneys, very, very large, and these are smaller kidneys with much fewer cysts. And seem to be tolerated and safe in mice. And again, people have been taking this drug for many years for the purposes of lowering cholesterol. So we now have a phase 2 trial. We are very gracious. We are very grateful that this has been funded by the Department of Defense, which has been a key federal funder of many of these clinical trials in polycystic kidney disease. And thanks to the lobbying by the PKD Foundation for that funding to be continually supported. It is a placebo-controlled. So again, people are equally likely to get the active drug or an inert drug. It's double-blinded, which means neither a participant nor us, as the doctors will know which one we're getting, and that's to maintain the blinding. So we have 3 clinical sites, us here at Maryland and University of Vermont and Tufts University. So if you have friends, colleagues, family members who live in Boston and Northern New England who are looking for opportunities to take part, we can direct you there. It's looking at this drug taken just once a day. It's a pill, and it's over a 2-year period. So the kinds of patients that would be eligible for the study are 18 to 60 years old, and they're at high risk for progression based on that Mayo imaging classification scheme that I provided a little while ago. Unlike most of the drug trials that I'm talking about today, in fact, every other drug trial I'm talking about today, patients who are using tolvaptan are eligible for this study. So this would be add-on therapy for them. Or if you're not using tolvaptan, you're still eligible as long as your kidney function is at least 35, because this drug is only approved for people who have kidney function over 30. If you're on tolvaptan, we just ask that you're not being recently started on it and you're on a stable dose and tolerating it. And so the sort of schema of and I'll give some more detail on this. The schema of the trial and what it kind of looks like is shown here. So patients are assigned to either placebo or active drug. We have a screening or baseline visit, and then there's some safety visits for labs. We call them on the phone. And then the last visit is at 24 months. So some of these are in person, some of these are lab. I'll show it right here. There are 7 in-person visits over 2 years, and that includes the initial screening visit to determine if someone's eligible. 5 remote visits. You go to LabCorp, someone goes to LabCorp, then we call you on the phone, talk to see how the person's doing. 3 abdominal MRI scans. And again, if someone is taking tolvaptan, they can be eligible for the study as well. So that study we've been working on now for about 6 months. So we're just about ready to start enrolling in it within the next couple months. We're just waiting for the study drug to be manufactured and available. Another drug that you've probably heard a lot about, be impossible not to, is the GLP-1 receptor antagonist. So what are these? GLP is a protein released normally after meal consumption, and it regulates sugar metabolism, satiety, that's fullness, and intestinal function. So the drugs that block this receptor of this protein induce weight loss, and they're approved for use in type 2 diabetes, obesity, now in heart failure. So Ozempic, Mounjaro, what's the other one? Rybelsus, these are all, right? You can't, again, watch TV without seeing these advertised. So it turns out that they also work in kidney diseases, in this case, diabetic kidney disease, where they lower the risk of kidney failure and chronic kidney disease progression. And here on this figure, the y-axis is the number of people who have gone on to kidney failure after starting the trial. The black line is the placebo, and the orange or brown line is the people randomized to Ozempic. So you can see at every time point, there's far fewer people who got Ozempic that ended up with kidney failure, actually about 25% less. So this drug is actually FDA approved, but only for diabetic kidney disease currently for the treatment of it. So what does it do in polycystic kidney disease? There is an ongoing phase 2 trial. We are not part of the trial here, but I did want to make people aware of it. Our colleague at the University of Colorado, who has federal funding for this trial, and she is actually using not Ozempic, but a drug in the same exact class, which is called, in this case, Zepbound. So you've probably heard about it as well. Okay. I did want to move on now to the other drugs that are not being repurposed, and we heard a lot about this one from Dr. Germino. So this is farabursen, and as I think he very nicely described with that cool cartoon and animation, it's an oligonucleotide, which blocks this mRNA, which helps to regulate the production of polycystin proteins. This is a drug that's administered by injection under the skin. And so again, it increases the production of polycystin, and in mice, it slows the rate of cyst enlargement and results in better kidney function compared to placebo. And this is just one example of a whole range of mouse studies that were done using different mouse models, that is, different ways to genetically knock out or knock down the function of polycystic kidney of PKD genes. This is the mouse that got just the vehicle, that is placebo. This is the mouse that got farabursen, and you can see, yes, of course, there are still some cysts, but it's markedly less cystic and markedly smaller, right? That is better. This is the phase 3 trial that is going on. Let me just, as a precursor to that, it has been studied in an earlier phase, what we call a phase 1b trial. We were one of the sites that participated in it. And they gave different doses of the drug, and they found that it increases the amount of functioning polycystin protein in the urine, suggesting, again, that there's more production in the kidney. And there was a suggestion that the growth in kidney size was slower compared to placebo. So this drug was originally developed by Regulus, a small biotech company. It was then bought by Novartis, the large multinational company, for several $billion. Obviously, they would only put the money in if they thought there was some promise to it. But of course, we still have to test to see in the long term whether it is effective at the clinically relevant outcome of slowing kidney enlargement and improving or slowing kidney function decline. And that's exactly what this trial is doing. So it's going to look at the effects of kidney enlargement over 12 months and on kidney function over 24 months. There's randomly assigned to either the placebo or one of 2 doses of farabursen. So you actually, more people will get the active drug than placebo, you're more likely. And it's given by injection every 2 weeks. So people who are eligible for this study are at age 18 to 70 and again have large kidneys for age. They're at high risk for progression. And we are one of the sites that will be participating in it. And at the end, give some information to contact us. So we're in the process still of contracting. This trial has not started enrolling yet, but will be soon. We heard also about this drug from Vertex. This is their trial that they're looking at, I think has the acronym AGLOW. I'm sorry, I don't know what that stands for, the acronym. It's kind of cool, but it's a novel experimental drug. This is an oral drug, a pill, that targets the misfolding, as you heard from Dr. Germino, of polycystin 1. And this is a new kind of drug and a new kind of trial because it's a drug specifically designed for specific types of mutations. So it's a genetically based drug and a genetically based trial. These are the missense mutations, the kinds that Dr. Germino was describing, where there still is protein being produced, but the protein isn't folding right and it isn't getting to the correct location in the cell. So this trial is starting enrollment. We're in the process of getting it onboarded here. I apologize. We are actively enrolling here. I should clarify. It's going to look at the effect of the drug on kidney growth over 1 year. So you have to have one of these kinds of mutations, and people who are eligible are 18 to 65 and have kidney function of at least 25 mLs per minute. One of the interesting features of this, of course, a lot of people don't know their genotype because genotyping is kind of expensive and a lot of times it's hard to come by. So this study will actually pay you to get genotyped. So if someone doesn't know their genetic mutation, they can come into our research center or others, and the company will pay for your genotyping. And even if you're ineligible, of course, you'll still have that information on genotyping. So if someone is thinking about this or might be potentially interested, but doesn't know if they meet the criteria based on genetics, that can be handled and at no cost. Okay. We heard also from Dr. Germino about this new target, PAPA or papalysin, and its relationship with growth factors which regulate or promote the proliferation of cells abnormally in polycystic kidney disease, and particularly a growth factor called IGF, or insulin-like growth factor 1. Insulin-like growth factor 1 is itself bound to different proteins which prevent the growth factor from reaching the target, its own cells. And then one level of control beyond that is papalysin, or PAPA-A, which, as Dr. Germino described, was discovered originally in pregnant women. And this clips the binding proteins, thereby permitting more growth factor to get to the kidney cell and promote cyst expansion. I'm going to show a cartoon that makes more sense of this in a second. So PAPA-A, the clipping enzyme, is increased in polycystic disease, and it actually is higher in worsening disease. And that leads to greater binding of IGF-1 to its cells and more cysts. So IGF-1, the growth factor, is in here. It binds to its receptor. It leads to more cysts. Normally, it's bound to this binding protein, so it can't get to its target. It can't create more cysts. But this clipping enzyme will clip the binding protein, freeing more. So the idea is, what happens if we block this, keep more growth factor bound to its binding protein, therefore it can't get to the cells and the kidneys, it can't trigger more cysts. In fact, that's exactly what happens. This is what happens when you take away the gene for papalysin. This is a normal kidney. This is a kidney with polycystic disease, and this is a kidney with polycystic disease in which there is no papalysin around. So it looks just like a normal kidney. But that's not really practical, obviously, for human beings. We don't want to obliterate a gene that has some important function. So instead, we can actually just inhibit the protein papalysin by giving a drug, an antibody. And that works as well, and that's shown here. This is the treated mouse. This is the cystic mouse without treatment. And this is just ways to quantify that the kidneys are much smaller after treatment. So there are 2 drugs that are already being starting enrollment or starting testing. And I'm going to go through those briefly before we wrap up. One is made by a company called AbbVie in collaboration with another company called Calico. So these are monoclonal antibodies. So these were engineered to target this PAPA protein. This is a phase 2 clinical trial. The drug is given intravenously. So this is not a drug that could be, so far at least, administered at home. It's administered monthly for just under 2 years. You get either placebo or one of 3 doses. So they're trying to figure out what's the right dose of this drug. And they're going to enroll 240 people worldwide. Again, you have to be at high risk for progression. And it's going to look at its effects on kidney volumes over about a 2-year period. So this is, I'm sorry, I left out the acronym for this trial, but this is the drug made by AbbVie. And this is the drug made by AstraZeneca. And their trial is an acronym PIONEER. It's a slightly earlier phase trial, so it's not looking at 2 years or effects, but a much shorter period of effect. It will only enroll 40 people. They have to be between the ages of 18 and 65, have large kidneys for age. One important thing about this drug and this trial, and I'm sorry, I don't know, I didn't comment on it for the other drug in the same class, because of the effects on the developing fetus are unknown. Women who enroll in this trial must be of non-childbearing potential. That would be after menopause or after tubal ligation or hysterectomy. Again, we're giving it by injection, and 3/4 of the people will be randomized to active drug, 1/4 to placebo. This is an earlier phase trial, so we're trying to understand how the drug is absorbed in the body, how it's excreted, how it's metabolized. And so it does involve several overnight stays in our research unit in our hospital. Those are obviously fully supported and reimbursed for, and in addition, a large stipend for reimbursement for participants. And then lastly, just to point you out to the PKD patient registry, which is a wonderful national project by the PKD Foundation that Dr. Wadnick and I have also been involved in collaborating and leading. And the idea is to store information about patients that can be used to help understand the progression of disease. It's fully voluntary, of course. All the information is de-identified to protect the confidentiality. And also it is a way to also help investigators understand how many people might be eligible for certain kinds of trials. And then lastly, within the PKD Foundation's website, I just wanted to point everyone out that this is a wonderful resource to help understand, here's where I live, here's a little bit about me, what kinds of opportunities to take parts in clinical trials are available. And that might be at our institution or that might be at other institutions. And I just encourage people who are thinking about this to reach out to that. Our contact information is here. Rachel, you already know. Charlotte Diggs, you're going to be hearing from later, our wonderful research coordinator. And thank you very much for your time.
Steve, let me ask this. Does bempedoic acid also benefit polycystic liver disease cysts?
Right. It's an excellent question, and we're hypothesizing that it will. So most of the mouse models where we induce polycystic kidney disease, it doesn't affect the liver. So this hasn't been studied thus far in experimental models. We will be looking at that very question, though. In this research study, we're going to be quantifying the cystic involvement in the liver in people who get the study drug and compared it to placebo.
Okay, so is anyone going to be told, do we know, talking about diet? Yes, in a later talk, we will be speaking about diet. Okay, do post-transplant patients qualify for the AGLOW study if other?
No, it's an excellent question, and I'm pretty certain that all these clinical trials will exclude patients with prior transplant. Yeah, and there's complicated reasons for that. So these are all trials that are designed for people with native kidney disease. But we think that there's still a lot of people who are potentially eligible for those trials. Thanks.
Anyone here have a question? I'll just take a quick, there was one question from Dr. Germino's talk. If protein is too low, why are patients advised not to have much protein in their diet? So what I think Dr. Germino was getting at is that the PKD1, the amount of active protein is too low, not talking about all proteins in the body. I don't know, Steve, if you want to comment.
Yeah, I think that's right. And we'll defer to the dietary discussion about recommendations for protein intake. But we're really talking about very specifically changes in one protein that represents a tiny infinitesimal speck of the total protein circulating. And even then, we're talking about how that protein is produced inside the kidney, not circulating through blood. Yeah.
One more in the chat. Okay. Okay, is being on a GLP-1 drug an exclusion criteria for participating in the faribursin, AGLOW, or PAPA?
That's an excellent question. I am not aware that it is, and it is not for our bempedoic acid trial. So, I mean, it's interesting. I think companies will have to kind of address this because so many people are using these drugs not to treat their polycystic kidney disease, but to treat overweight and obesity, and they have enormous benefits for that. But I am not aware that, at least to the best of my knowledge, that those are exclusionary criteria. But certainly, if you have interest and if you're near us, just contact us. We can go through that in detail. And if there's uncertainty still, we can then query the companies. Thanks. Great question.
Okay, so with that, Rachel, is it a break time? Okay. Oh, Paul. Oh, let me.
Sorry.
I just wanted to mention, in addition to our registry, and thanks for mentioning the registry, we also have our accelerating clinical trial alerts, ACT alerts. We got about 15,000 people now on our ACT alerts. Again, for those of you who are remote, I'm Paul Scribner from the foundation, PKD Foundation. So if you want to make sure you hear about the newest opportunities for participating in clinical trials at any level, we're regularly getting requests for everything from ad boards, an early trial development, all the way through actual participation in sort of phase 1, 2, and phase 3 trials. So make sure you're on those alerts. We send out emails regularly.
Yeah, thank you. And I would say, just as a sort of larger framing of this, been involved in this field and for 15 years, this is by far the most exciting time in terms of the number of drugs that are being developed and going to testing. And Dr. Wadnick and I, it's probably not a month that goes by without getting approached by some company, maybe a small biopharma company, interested in testing and developing their drugs. So I think there's a lot of reasons to be very hopeful and optimistic. Thanks.
So we're going to take a break until 11:30. Well, it's to 11:24. So we have a few minutes for people here to have a bio break and have something to eat. So thank you, everybody.